So the composite site lability is one factor we can influence to improve the metabolic stability, but I mentioned before that it is not itself a prediction of the rate of metabolism. Why is this? Well, the P450 catalytic cycle is very complex and actually has many potentially rate-limiting steps. So what this means is in practice that quantitative prediction of metabolic rate, whether that be intrinsic clearance or half-life, for a diverse chemistry, for a global data set is intractable based on structure alone. So we have to consider, for example, binding. The Km in Michaelis-Menten compound doesn't bind. It doesn't matter how labile it is. It won't be rapidly metabolized. The reduction steps can be rate limiting, particularly influenced by type 1 and type 2 binding. And the decoupling pathways can also reduce that rate of metabolism significantly. Whereas the composite site lability is looking at one very important factor, which is the efficiency of this product formation step. However, what's good news is that it is certainly possible to build local models of metabolic rate. That's because within a given chemical series or project's chemistry, it's likely that only a small number, one or two of those factors, will be very influential on the overall rate of metabolism. So let's look at an example of this. This was an example where we used the auto modeler in our StarDrop software to build a model of the rate of metabolism by CYP3A4. This was expressed as a log of the rate constant. The model was built based on a 100 compound data set from a project. The descriptors we used were indeed that composite site lability that captures that efficiency of metabolism, but also two factors that are very important for determining the binding affinity, the log P, and the polarity of the compound. In this case, we're looking at the log of the neutral fraction at physiological pH. And what you can see here for the independent validation and test sets is a really very good correlation with a coefficient of determination of almost 0.7. And just looking at the specificity of the model, particularly what you can see is of those compounds that were predicted to meet the stability criteria, the large majority of those were found to be stable in practice in those independent test sets. So this was then applied to a virtual library of 13,000 compounds designed by the chemists in this project. From that 13,000, 40 compounds were selected for predicted stability and potency. And from these 40 compounds, 4 new potent and stable series were identified, so illustrated here. All 4 of these series, A, B, C, and D, are identified to have highly active compounds and to meet the metabolic stability criteria, in this case, for this project, of greater than 20-minute half-life in vitro. So within these local contexts, one can make highly quantitative and accurate predictions of the rate of metabolism.